Welcome to my channel, Correct Biology. In this video, I am going to teach you about Mendel work, and here we are going to discuss monohybrid cross. So here now we are going to start the discussion related to the work done by G.J. Mendel. And hopefully you remember G.J. Mendel. He is well known as father of genetics. So today we are going to discuss the work done by G.J. Mendel, and initially we are going to discuss monohybrid cross. Today in this video you are going to learn two laws. One is law of dominance and second law of segregation. Okay, so be with me and learn the Mendelian work, that is monohybrid cross. Now here I just want to tell you G.J. Mendel, okay, keep in mind he is known as father of genetics. Okay, remember he is known as father of genetics. Why? Actually, because due to his work this field started growing. Okay, and here hopefully you remember G.J. Mendel, he carried out work on pea plant, okay, by some sativum. Okay, so this is pea plant. Now here question is sometime being asked, why Mendel choose pea plant? Okay, so here there are some simple answer. One is it is very easy to grow. Okay, you don't need special expertise, you can easily grow the pea plant. Second, here what you are going to see, it is quite easy to perform self-fertilization. Okay, now here I just want to tell you, if you see the flower of this plant, it is going to have female part and it is also going to have the male part. So male part and female part, okay, both are in a single flower. Okay, so here you can see that within a single flower you are going to have both male part and female part, and naturally what you are going to see here, you are going to see self-fertilization. Yes, experimentally you can also carry out cross fertilization. Okay, cross fertilization. Now how you can carry out cross fertilization in the pea plant? Just let us draw this structure again. So here I am drawing that this plant is going to have female part and this is going to also going to have male part. Okay, so this is, let's suppose these are anthers. Okay, this is stigma style ovary. Okay, the female part, and these are the male part. Okay, these are present within the one flower. Okay, now try to see here. Mendel carried out cross fertilization. Okay, how? When this flower, it is juvenile. Okay, this flower is not matured. Okay, let's suppose this is the female part, not well developed. This is the male part, even that is not developed. Okay, what Mendel did, he cut and removed the male part. Okay, now what happened? So when this flower it get matured, okay, it was having only the female part, okay, and not the male part. Okay, so this is, you have to keep in mind how Mendel carried out the cross fertilization. Now here this flower is only going to have female part, and now here you can collect stamens from this. Okay, so you can collect stamens from this with the help of a brush, and just by touching the female part, okay, you can perform the cross fertilization. So keep in mind, Mendel, he performed two type of cross. One is self-fertilization. Self-fertilization in this plant, it is going to occur, 1 minute please. Self-fertilization, it occurred naturally, okay, and I have told you that you can here also perform cross fertilization, okay. So hopefully it is clear. Next here I just want to tell you, this pea plant, it reproduce well and grows in a single season. Try to see here, it reproduces well, okay. When you are going to see after fertilization, what you are going to see, the next generation you are going to see, the number of offsprings you are going to see, okay, there are many, okay, you can have a large number of data, okay. So this was also good. Second, it grows in a single season, okay, you have not to wait, okay, for years, okay, to get the result. So within a single season, Mendel used to get the results. Now another important thing here, what is you are going to see, that Mendel, he choose 7 contrasting characters, okay. So 7 easily visible contrasting phenotype. So hopefully you understand what is phenotype. It is the external appearance of the plant, okay. So here what we are going to see, 7 contrasting characters, means you can easily distinguish, and you can easily see different phenotypes. Try to see, Mendel, when he carried out this experiment on the pea plant, he was not having any special type of instruments. So he has to believe on his naked eye, and by naked eye he was able to see 7 different contrasting characters in this plants. That's why he chose this pea plant. So here let us discuss the 7 contrasting characters which were being chosen by Mendel, okay. So let's see what are the characters, okay. First is the character, he chose the height, okay. So here this character height, it can have 2 contrasting traits, okay. So here height can be tall, it can be dwarf. Second, here you can see the flower color. Here the flower color can be violet, or you can have white. Third, here you are going to have flower position, okay. So the flower position can be axial, or you can have terminal, okay. So here I just want to tell you, in case of terminal, what we are going to see, let's suppose this is the plant, okay. So it is going to have different branches, okay. So here at the top, okay, if you are going to have all the flowers, so this is the terminal position of the flower, okay. And when you are going to have flowers, okay, coming out of these nodes, okay, so this is your axial position. So here. So hopefully it is clear to you what is the axial positions. Okay. Next here, I just want to tell you, in this plant, the another character what you can see, the seed, okay, color, okay, it can be yellow and green, okay. Then here you are going to see the seed shape, okay, it can be round or it can be wrinkled. Next here if you see the sixth one, here you can see bold color, okay, it can be green or it can be yellow. Next here the seventh one, okay, here you can see the pod shape, okay, it can be full or it can be constricted, okay. Here you can say full or you can say smooth, okay. So the shape of the pod, it can be smooth or it is going to be have some rough surface that is constricted. So try to see, Mendel, he worked on a pea plant, and this plant he saw, within this plant he saw 7 visible characters, and all these characters, they were having contrasting phenotypes, okay, contrasting traits. Now let us discuss how actually Mendel carried out the experiment. This is important, very important. Keep in mind, Mendel, okay, he carried out the experiment using true breeding varieties or strains. Okay, now try to understand what is this true breeding variety. Now initially, okay, what he did, he crossed true, okay, so here, sorry, tall, let's suppose tall cross with tall, okay. And what you are going to see when you are going to have a tall plant crossed with tall plant, I have told you how you can perform the cross fertilization. What he got, okay, tall, yes, few dwarf, okay, few dwarf, okay. Here we don't know whether it is true breeding or not. So this tall was selected, okay. Again it was being crossed with tall until at the last you are only getting tall. This is important. Try to see here, this tall, okay, it can be homozygous or heterozygous, but this one is homozygous, okay. Try to see here, okay. Today we know that tall plant, it can have two type of genotype. It can have capital T, or it can have capital T, small t, okay. Okay, now these are going to represent the allele. These are going to represent the allele. What are alleles? Alternative form of genes. So in a tall plant, you can have capital T, small t. Now here, if you see, this is homozygous condition and this is heterozygous condition. In homozygous condition, both allele are same. In heterozygous condition, both allele are not same. Now this homozygous, okay, plant, okay, this is actually the true breeding. It will always give rise to tall, okay. So try to think how actually Mendel performed the cross by initially selecting the true breeding. And how he selected? He crossed tall with tall, okay, until he was having only the tall. Okay, so first he initially collected seeds from the true breeding varieties, then he performed the cross. Okay, so now we are going to discuss this monohybrid cross. Okay, now try to see here what is this mono. Mono means single. Okay, hybrid here stand for hybridization. In monohybrid cross, what we are going to have, you are going to consider one pair of contrasting trait. So any cross done by taking one pair of contrasting trait, it is known as monohybrid cross. So here let us see, Mendel started with tall plant crossed with dwarf. Okay, so here I have told you how you can perform the cross fertilization. So in the case of tall plant, okay, let's suppose here he removed the male part, only female part is there, okay, and he removed the anthers at the juvenile stage, and later he pollinate this with the dwarf. Okay, you can do in other way also, but here let's take the for simplicity, let's tall plant crossed with dwarf. So initially he performed the cross fertilization. So here I have told you he started the experiment with true breeding variety. So tall. So you have to take the genotype capital T, crossed with small t. Okay, so keep in mind, Mendel, he started the experiment using the true breeding varieties. In the case of true breeding varieties, you are going to have the same genotype. And now, what he got the result in F1 generation, okay, this is F1 generation, what he got, all tall. So here, how you can solve this? So what he did, tall cross with dwarf. Here what we are going to see, you are going to see formation of gamete, and here only one type of gamete, here also one type of gamete. Okay, capital T, small t. Now when they are going to fuse, fertilize, what you are going to see, the genotype capital T, small t, and this is your F1 generation, and here this is your parental 1 generation. Okay, these are your gametes. Okay, now here you can see whenever we are going to form gametes, we are going to circle it. So this is in the first generation, what he saw, all tall. Now here I just want to tell you, you can see that this is going to reject earlier hypothesis. Now try to see, before Mendel there was one hypothesis that characters, they are being passed from one generation to next generation and they are being blended. So earlier there was a theory, that is blending hypothesis. As per blending hypothesis, the next progeny, they are going to have intermediate, they are going to have mixture of the characters from both of the parents. But what here Mendel saw, that when tall plant, it is being crossed with dwarf, he did not saw intermediate. What he saw? All tall. Okay. Next here, what he performed, the self-fertilization. He allowed the self-fertilization of all the F1 generation plant. Okay, what he did, he allowed the self-fertilization. Okay, so initially here cross fertilization, and here what he allowed? Self-fertilization. Okay, so capital T small t cross with capital T small t. So here what he is allowing? Self-fertilization. Now here also what you are going to see? Let's see. 1 minute please, some technical error. Why it is not having? Yes. Okay, so here you see capital T small t cross with capital T small t. Now here what you are going to see? There is going to be formation of two type of gametes, capital T small t. Here also you are going to see formation of two type of gametes, capital T small t. Now if you solve this by Punnett square, you can easily solve. Okay, capital T small t, okay, capital T small t. Now I just want to tell you what Mendel got the result. So here Mendel, okay, when he performed the cross, okay, what he got? 787 tall and 277 dwarf. Okay, so this was the actual result, and if you solve this ratio will come out 2.84:1. Now here if you solve this cross by using a Punnett square, so here capital T, then you are going to have this, then here you see capital T, then 1 minute please, you are going to have small t. Okay, so capital T small t. Now here if you see this one, it is going to have so this genotype in the homozygous condition, okay, you are going to have tall, here you are going to have again tall, okay, here also you are going to have tall, and here you are going to have dwarf. Okay, now try to see here that here what you are going to see, phenotypic ratio 3:1. Okay, at 3 place what you got? Tall, at 1 place dwarf. Now this is the Mendelian cross, and here from monohybrid cross, okay, you are going to learn two laws. One, here if you remember that when tall was crossed with dwarf, okay, capital T crossed with small t, what you get? Okay, what he get? Capital T small t tall. Now try to see here. So Mendel, he was not having any idea of chromosomes, he was not having any idea of the process of meiosis. Okay, he thought that for every trait, okay, for every character, okay, for each character, okay, there are 2 unit factors. For each characters there are 2 unit factors. Okay, so let's suppose this is the height is the character, and for controlling the height there are 2 unit factors, and the factor which is dominant, okay, that will be expressed in the F1 generation. The dominant factor is expressed in F1 generation. So here you see there were 2 alleles, okay. Now out of these 2 unit factors, okay, one will be expressed. So this is going to be expressed, and the one which is being expressed in the F1 generation, that is the dominant factor. The another one is recessive factor. Okay, so here you can easily in a simple way say this, that whenever you are going to perform cross, okay, so what you are going to have? You are going to have two unit factors, and the dominant factor will be expressed while the recessive factor will not be expressed in the F1 generation. This is known as law of dominance. Law of dominance. Okay. So here in a simple word, what you can say? As per law of dominance, okay, the dominant factor will be expressed in F1 generation. Dominant one. Now here try to see. We are going to have this cell. Okay, let's suppose you are going to have just two chromosomes. Okay, the one pair we are going to see. So here what we are going to have, capital T factor is there, small t is there. Okay, in this situation, okay, so one minute please, let's take it both capital T, capital T. Okay, now in this situation, what is going to happen? This plant, okay, it is going to produce proteins. Okay, these factors, today we know these are now known as genes. These factors, they are going to code for proteins, and these factors, when they are going to code for proteins, yes, there may be some enzymes, and these proteins or enzymes, they are carrying out work. They are going to carry out biochemical reactions within the plant. The end result is the height of the plant is tall. Okay, same way here, if you are going to have situation when you are going to have capital T and small t, in this condition, what you are going to see? You are going to see synthesis of proteins, enzymes, and the end result is the plant is going to have the height tall. But if you are going to have the situation when you are going to have small t, what you are going to see? You are going to see no synthesis of this protein, or you are going to have something truncated. So there is some mutation. Try to see here these factors. Okay, these factors today we know as genes. And remember, alternative form of genes. Remember here, alternative forms of genes present at same locus. They are known as alleles. So here, if you see the height, it is being controlled by two factors. One is capital T, another is small t. Okay, so these are actually genes. Okay, one is capital T and another is small t. So here where you are going to have capital T, so let's suppose it is going to form some proteins. Okay, but this one, it is mutated. Okay, this is not going to form the desired protein, and the result is that you are not going to see the expected result. So hopefully it is clear to you what is monohybrid cross and what is the law of dominance. Keep in mind the dominant factor will be expressed in the F1 generation. Now here from monohybrid cross, you also learn there is another law you can also learn, that is known as law of segregation. Okay, keep in mind this law of segregation, it is also known as law of purity of gametes. Try to see here, during the time of gamete formation, the unit factor segregate. Okay, try to see here when we perform self-fertilization, okay, capital T, small t, okay, these were present within the genotype, but during the time of gamete formation, they segregate. Capital T and small t, they segregate, and they were present in different gametes. So here from Mendelian monohybrid cross, what you have seen that you can learn two laws. One is law of dominance, another is law of segregation. Now here I just want to tell you that from Mendelian monohybrid cross, okay, if you remember the Punnett square, okay, capital T, capital T, capital T, small t, capital T, small t, small t, small t. Okay, so here this was tall, this was tall, this was tall, and this was dwarf. Okay, so here what you get, phenotypic ratio 3:1, okay, at 3 place tall, at 1 place dwarf. So here phenotypic ratio 3:1. And if you see what is going to be the genotypic ratio, so genotype, how many genotypes? This capital T, one time, capital T, small t, okay. So if you see at this place, okay, so here 2 times, and this small t, 1 time. So here the genotypic ratio is 1:2:1. So keep in mind, if you are going to solve monohybrid cross, the genotypic ratio 3:1 and 1:2:1. Now here I have shown you for one character height, and I have told you Mendel, he performed the same cross with 7, and in all the cross what he got, this ratio 3:1 ratio. So hopefully you have understood monohybrid cross. Thank you for watching this. Please do like, share the video, okay, and subscribe to my channel Correct Biology. Thank you.